Modern biology frequently relies on a mechanical assumption that living organisms are bottom-up assemblies of passive molecular parts. In this model, DNA is the hard-coded blueprint, a master set of instructions that dictates every physical outcome. This view frames the genome as a static, physical schematic. It suggests a one-to-one mapping between a set of genes and the final structure of the organism. However, this rigid model fails to explain how cells navigate entirely new environments. Empirical evidence from regenerative medicine shows that biological systems can solve complex structural problems and build novel forms that their genetic blueprints never explicitly detailed. Bridging the gap between these anomalies and classical physics requires a move toward informational ledger theory, which treats information as the primary substrate of reality. At the most foundational level, reality exists as a boundless configuration space, a pure probability field containing every possible computational rule and state in a condition of superposition. In this framework, DNA serves as a standardized set of hardware. The software driving physical form is the bioelectric information that allows cellular networks to communicate and process their environment. Anatomy, therefore, appears to be an emergent computational interface, a structure compiled by goal-directed biological materials that actively resolve uncertainty in the information network. If anatomy is an interface, it changes how we view macroevolutionary transitions, the moments when entirely new body plans emerge over millions of years. A common misconception suggests that nature must manufacture entirely new genetic material from scratch to generate these morphological changes. Evolutionary developmental biology reveals a different process. When we analyze simple organisms like the fruit fly, we find evidence for deep homology. Species as diverse as crabs, insects, and humans share a foundational toolkit of approximately 14,000 genes. These genes are preserved across hundreds of millions of years of evolution. This modular genetic network operates as a collective system. Speciation happens when this same exact gene set alters its connection software. Rerouting these interacting nodes projects a radically different anatomical feature, like a beetle horn, without needing new genes. This process is known as co-option. Evolution functions as a software engineer, rewiring existing materials to serve new purposes rather than inventing new parts. Because morphological changes rely on reprogramming these connections, DNA is best understood as flexible hardware rather than a fixed schematic. If DNA provides the hardware, what actually computes the final anatomical instructions? The driver is multi-scale competency, the inherent intelligence and problem-solving ability that exists within individual cells and molecular networks. Anthrobots provide a clear example of this. These are created from adult human tracheal cells, which are liberated from the body's standard instructive signals in a laboratory setting. Without any genetic modification, these independent cells self-assemble and begin to locomote through their environment as a coherent agent. In these experiments, the anthrobots exhibit a specialized behavior. They can locate and physically knit together damaged human neural tissue. Because tracheal cells were never exposed to neural injury during evolution, this healing behavior is a form of zero-shot learning. The cells are solving a novel structural problem in real time. These cells are accessing an unmapped mathematical space of potential forms. This morphological latent space is a Platonic realm of anatomical possibilities. It contains alternative structural outcomes that standard wild-type hardware can access and stabilize. The genome does not dictate a final form. It provides the materials for cellular agents to explore and compile a wide range of morphological potentials. This raises the question of control. How do cellular collectives communicate and store their structural targets? They utilize bioelectric networks as a form of active memory. This electrical signaling layer serves as the primary software for setting structural goals. We can observe this bioelectric layer in the planarian flatworm, a creature capable of regrowing its entire body from small fragments. A false color scan shows the flatworm's bioelectric memory map, where specific head and tail voltages template regrowth. Researchers can overwrite this state, changing the tail's electrical signature to mirror the head. Following this new target, the collective physically constructs a two-headed flatworm. The underlying DNA, the physical hardware, remains wild-type. However, the software state has been permanently altered to a counterfactual memory. These bioelectric targets prove that anatomy is explicitly programmable. Physical form is a software consensus rather than a genetic mandate. The goal-directed agency observed in these biological cells suggests a deeper alignment with the laws of information. As biological matter functions as a programmable interface, it implies the universe itself may operate as an informational substrate. Informational ledger theory analyzes the early universe as a probability field, where specific computational rules, or chiplets, spontaneously nucleate and stabilize. These systems naturally build toward stability by minimizing informational entropy and predictive uncertainty, following the free energy principle. This drive for efficiency is explained by Donald Hoffman's interface theory, specifically the mathematical theorem that fitness beats truth. Evolutionary selection inherently punishes systems attempting to render absolute reality. Algorithms optimized for survival fitness consistently outcompete those estimating the objective world. Perception favors survival utility and data compression, suggesting the objects we see are simplified icons, not foundational truths. Space, time, and biological forms represent the user interface of an underlying quantum computational ledger striving for maximum predictive certainty. Integrating these biological insights with computational physics points towards a specific horizon, the anatomical compiler. We are shifting from micromanaging low-level genetic pathways to communicating high-level abstract structural goals directly to cellular collectives. This compiler functions as an AI discovery engine. It proposes a bioelectric stimulus, observes cell behavior, and updates a predictive model. This feedback loop bypasses slow evolution, using optimized stimuli to instantly access new attractors in the morphological latent space. The biological world functions as an actively self-compiling entity, executing a relentless optimization algorithm to resolve its own uncertainty. This framework unifies quantum computational physics, cognitive science, and synthetic morphology into a single, cohesive architecture. The era of viewing biology as a collection of rigid mechanical gears has ended. By speaking the software language of the bioelectric ledger, we gain the ability to compile and restructure reality itself.